Hello, in this video we will discuss about the transplantation rejection. After the transplantation rejection, how the process will occur. So let's begin to understand. This is the person which that is the healthy person and it is donor and it will donate the kidney, lungs, liver part. So that kidney transplantation, liver transplantation or lungs transplantation to the kidney failure person or lung liver person is the recipient. So this is a transplantation. After the transplantation will occur, it means the cells of the healthy person will introduce, for example, liver cell, kidney cell, and this is the grafted tissue. It is known as the grafted tissue. After this antigen will introduce, this is the basically antigen. If it will mismatch, we will discuss in the another video for cross match process. So this is the basically is the antigen grafted tissue as a it will be graft the products shed from graft due to the surgical stress or by uptake by professional antigen presenting cell. So the antigen presenting antigen processing and presentation via a uptake of antigen of the grafted tissue as antigen. And let's begin to understand the process how it works this antigen presenting cell. So this is the phagocytosis will be occur of this antigen, the part of antigen, and go inside is known as endosome formation, and this endosome will be fused with the endoplasmic reticulum. After the endoplasmic reticulum combining, the invariant chain which that is contain receptor will move toward the endoplasmic reticulum, which basically are present in the endoplasmic reticulum and go into the after the combining with the endosome. So here you can see, and in this way it will invariant chain will be separate, and after this the antigen recognition will show with the plasma membrane. So MSC class 2 molecule which that will show the antigen for another and go into the lymphatic system. So remember, this is the lymphatic system contain B cell and T cell. So after this, naive T cell recognized through antigen representing cell will proliferate into the T helper 1 cell, pro-inflammatory, and T helper 2 cell is anti-inflammatory. So remember, the B cell is also present in the lymphatic system, but we will discuss later in this video. So it will produce the T helper 1 cell pro-inflammatory cytokines, interleukin 2, 12, and interferon gamma, and tumor necrotic factor alpha. And these pro-inflammatory cytokines basically go into the blood circulatory system, and T helper 1 cell is producing these cytokines. And these cytokines basically the blood circulatory system will enter the pro-inflammatory cytokines enter into the blood circulatory system, and the CD8 T cell, natural killer cell, and the macrophage activation via these pro-inflammatory cytokines. So this is the pro-inflammation. And after this, the CD8 T cytotoxic T cell, natural killer cell, and the T helper cell will activate these all cell. And in this way, the macrophage, which basically is upon activation, macrophage secrete a large amount of interleukin 12 to help maintain T helper 1 cell response. And macrophage also express the FC receptor and can therefore kill via antibody dependent cytotoxicity. So the AADC. So anyhow, in this way, here is the grafted tissue. Again, I am drawing for understanding. So the natural killer cell, this grafted tissue also produce cytokines. We recruit the natural killer cell, and this natural killer cell will side to the KI or receptor and in the killing induced receptor and the MSC class 1 molecule. So the natural killer cell we recognize in the acute phase, and this acute phase the cytolysis will be occur of this acute phase, and acute phase means the complete damage of the grafted tissue. While on the other hand, important thing here is the anti-inflammatory T helper 2 cell anti-inflammatory. This is against the inflammation and produce interleukin 4, 5 and 13, and this is the lymphatic system, remember, and this is basically a T helper 2 cell will inhibit the T helper 1 cell because anti-inflammatory, and interleukin 12 and interferon gamma produced to inhibit the T helper cell to the T helper cell 2. So in this way, remember that this is basically T helper 2 cell will anti-inflammatory. In this way the cytokines will produce, and in this way the anti-inflammation because inhibition of T helper 1 cell to recognize the antigen through a B cell via a antigen representing cell as a, and also cytokines will bind with the Fc receptor, cytokine receptor, and this is the chronic, and in this way chronic mean again and again and produce the B memory cell and plasma cell. This plasma cell produce a lot of antibodies against the grafted tissue, and the anti-inflammatory response is the humoral because humoral mean the antibody produce and opsonization of this grafted tissue, the graft enhances, and in this way you can understand this will be opsonization will recruit the macrophage, basophil and neutrophil, and this is basically anti-inflammatory is also against the graft because the graft is the antigen as antigen if the cross match will not complete, and the cross match will not occur. So that is why if the cross match will occur, it means the no and inflammation will occur. So in this way the CD8 T cell is also activated, and this is the pro-inflammatory cytokine will activate from the T helper 1 cell, and this is the pro-inflammatory response, adaptive response because the adaptive immune system, and also the B cell is also adaptive immune response, which that is the anti-inflammatory response, humoral. So this is the pro-inflammatory response, adaptive, which that is activated the cytotoxic T cell attack the graft endothelium and signal the apoptosis of cell via the release of granzyme and perforin. So in this way, let's begin to understand. This is the perforin we release to form a porine channel, and also the grain enzyme produce and hydrolytic also enzyme which that will kill the through a CD8 the cytotoxic T cell. After the killing, it means the apoptosis will be occur and the antigen MHC class 1 will recognize this. And in this way, you should need to understand the debris of the cell will remain behind. While this debris will release the tumor necrotic factor alpha and cytokines, the interleukin, and this is will lead to recruit the neutrophil, basophil, and this neutrophil basically engulf this debris. After this, will neutralize further through hydrolytic enzyme and lysozyme enzyme, and in this way the basophil also occur, and this is basically the macrophage we recruit after the engulfing through neutrophil. The erythrocytosis will be occur after the endocytosis from the neutrophil. So the erythrocytosis through macrophage for clearing the transplantation. So when cross match will not occur and the agglutination formation will occur during the cross match test. So if we will introduce this grafted tissue, so in this way it will increase the inflammation, will damage this antigen as a grafted tissue. So thanks for watching.